Hello, everyone, and welcome to our webinar on carrot breeding for organic production systems, important traits and CIOA advances by Dr. Philip Simon. This is the first webinar in what we're calling our carrot webinar double feature. The second one on carrot history and domestication and diversity will take place next month. These presentations present some of the findings of the Carrot Improvement for Organic Agriculture project funded by NIFA OREI. The presentation will last about 45 minutes, and then we'll have 30 minutes for audience questions. Today, I'm very pleased to welcome Philip Simon, who is a USDA Agricultural Research Service geneticist and a professor of horticulture at the University of Wisconsin-Madison. His research on carrot and garlic genetics and breeding is conducted to improve these crops for growers and consumers. He's particularly interested in carrot root knot nematode resistance, crop diversity and origins, and the nutritional quality and flavor of carrots and garlic. So welcome, Phil. I'm going to turn off my video here and you can take over.
Well, thanks very much. Thanks very much, Alice. I very much appreciate the opportunity to speak to you all today about some of the research work that we've had underway with the CIOA, or Carrot Improvement for Organic Egg project, as Alice has already outlined to some extent. So just to start things off, I want to, we include these slides at the end of the talk, but this project, the CIOA project, CIOA again stands for Carrot Improvement for Organic Agriculture. Project was initiated with USDA NIFA in 2011, and we've had ongoing funds for the project from that standpoint. Some of the work that I'll be talking about today was initiated before the CIOA project actually began or ran coincidentally with it, but contribute to the CIOA effort of improving carrots for organic agriculture. And those other funding sources include the California Fresh Carrot Advisory Board, USDA NIFA Specialty Crops, and of course my employer, USDA Ag Research Service. So another acknowledgement part of the talk I want to present up front, rather than save it all toward the end, is to note that this project, like all plant breeding, is really a very large team effort. It's certainly well beyond myself. And there's people within my management unit that work directly with me on a day-to-day basis, USDA ARS list here, and also some very significant cooperators at other institutions. And the key ones are listed here: the Organic Seed Alliance, University of California, both the ANR and well, primarily the ANR for this project, also UC Davis for some of the things, Washington State University, Purdue University, and University of Wisconsin. So again, just to point out that there's a very large team that has been involved in gathering the data and putting the story together that I'll be talking about today. So let's start in the beginning, and at least in the beginning of farming, with the first efforts of farmers of that in the case of crop improvement, it starts with crop domestication. And there was, with the domestication process, there were some advances in crop production that led to yield improvements and uniformity that go along with the domestication process. Listed here, things like crop recovery and improvements on crop utilization for baking, cooking, and eating as fresh or processed. At the same time, there are also improvements at the farming level, where evidence of first use of manure on farms, for instance, goes back about 4,500 years. So there's been a long process that underlies agriculture. I think we all realize that. And that's an important part of the story I'm telling, because the diversity of the crop, carrots in the case of what I'm going to be talking about, is critically important to what improvements we can make on the crop. And it turns out that, like other crops shown here, apples, beans, corn, and tomatoes, carrots in the upper left-hand part of this picture are very genetically diverse for their appearance, as you can tell in this image in this photo. And that diversity is a result of the whole domestication process and farming that's gone on before what we're talking about today. But that diversity is very critical and important in crop improvement. And that's what we looked at, that diversity as a significant part of the CIOA project. Um, and again, just one more background slide here to talk about some historical related things. But in the course of the last century, whereas the first agriculture by definition really was all organic, there's been a lot of displacement of organic production by more modern egg production methodologies. But in spite of that displacement of organic agriculture in the last century, the organic sector has been growing lately. And according to some 2018 statistics, about 4 to 5% of the food's coming from organic production, a significant part of fruit and vegetable production. And in case of carrots, the most recent accurate statistics go back to 2019, where about 1 carrot in 6, or 17% of the U.S. carrots, according to the USDA Ag statistics, is grown under organic management. And that's been going up since organic stats were taken, and because carrots is a relatively easy or doable crop to grow under organic management. Crop improvement for organic agriculture is of growing interest because the use of the carrot crop by organic growers is increasing. And that's what really led to the inception of the CIOA project, which, as I mentioned before, started in 2011. We've had two major parts to that project that I'll be talking about today. One is to develop new carrot breeding lines specifically targeted for organic production, at least as specific as it needs to be. And I'll be talking to you about how much of this is really specific for organic and versus overall of interest for carrot production. So carrot breeding is one of the first goals I'll talk about. I'll also mention in less detail the role of the root microbiome as it relates to carrot growth, which has been an ongoing part of the CIOA project since the project began. And as I go along, it'll become clear why the root microbiome has had some special attention in the project. So for starters, when we got the CIOA project going, and we've asked growers what are their interests and needs to improve the crop for organic production. There are some somewhat differences between organic production and conventional production. The most obvious ones are that the organic crop is grown with no conventional pesticides or conventional herbicides. And so that becomes a major issue to address when we're looking at carrot improvement for organic production. Along with that, the organic farms tend to be more diversified and variable in terms of things like soil, also in terms of where the carrot crop is grown, in comparison to the very large scale carrot production that is out there to backdrop the organic production. And that's where the soil nutrients come in, and that's actually where the root microbiome work comes in, because there can be very different soil nutrients available in organic production systems than what the typical conventional production system may be. In addition to those on-farm expectations or needs for carrot improvement by organic growers, you as the growers and often the immediate to the consumer deliverers of the carrot crop are more often confronting or interacting with the consumers of your products more directly than a lot of the large-scale carrot production. So your markets certainly can and do include very large markets that are covered by some of the big chain grocery stores, but many of you are selling carrots or providing carrots for local markets, and that translates to more direct consumer input and more interest along with that in novel, healthy food sources. And so these are the major considerations that I'll be talking about in some of the trait improvements for organic agriculture. And so when I'm talking about trait improvement, I'm talking about what have we done for taking that genetic diversity and improving the crop in terms of pest and disease resistance, weed competition, response to soil nutrients, and in needs for local markets and direct consumer contact. So given these attributes of organic agriculture, I'll be pointing out what's unique for organic production versus conventional. But the big question is there diversity there to address developing new disease resistance, new improved flavor, new interactions with soil nutrients? So the first question to project as what we might be successful with is whether genetic diversity is there to tap into. One point I'll mention toward the end of my talk is the necessity to actually select or grow the crop under organic practices to make advances in breeding carrots for organic production. So the main thing that I'll be talking about this genetic diversity to improve the crop, but I will say toward the end of my talk something about how essential it is to select under organic conditions. So going back to the list of main topics that we've addressed with the CIOA project, let's take a look at the pests and diseases that we have focused on first for improvement for organic agriculture. Of course, pests and diseases are important not only for organic production, but also for conventional, but they're certainly more critical because you don't have that opportunity to apply a pesticide if the pest appears. And so in order for us to make improvements for organic production, we have to be growing the carrot crop under organic production systems, at least organic from the standpoint of no use of pesticides, because obviously if you're breeding for nematode resistance in a field that's had a nematicide, you're not going to be able to make much progress. So that's pretty obvious, but I'll mention it just as part of how operationally we've gone forward with this particular part of the breeding project. And again, for both organic and conventional production, pest resistance continues to be one of the best ways to improve the crop and protect carrots from diseases. Not the only way, but often the best. And one thing I'll mention as I go along is how much published information is there out there to guide us with carrot improvement for pest and disease resistance for the breeding work. So just to answer the question of where do we start and what traits do we look at for carrot improvement for organic production, we started with a survey that initially was issued in 2014. We've had some follow-ups on it. The list remains the same. The top 3 diseases and pests that come up in surveys of carrot, organic, and conventional growers, as well as seed companies, processors, and with some input from consumer groups, is the top 3 pests are root knot nematodes, meaning breeding for nematode resistance, alternaria leaf blight, so we're looking for alternaria leaf blight resistance, and cavity spot, so looking for cavity spot resistance. So I'll talk about those 3 crops and the progress that we have made in improving those for the resistance of those 3 pests or diseases. So starting with root knot nematodes, the major root knot nematodes that show up in surveys and have been known actually for quite a while are the 2 first nematode species listed here, Meloidogyne incognita and Meloidogyne javanica, which are primarily for warm climate growth of carrots. That's where these warm season or warm climate nematodes primarily occurred. More recently, we have been also looking at resistance to carrots for Meloidogyne haploid resistance, where that particular nematode is much more prevalent in cooler soils. So in order to do the resistance testing, we looked at a diverse range of genetic sources to find sources of resistance. And in most cases, most of what we would see are carrots like the ones you see on the right of this image, where this happens to be a picture of the grow out of the cultivar Brasilia, grown in Meloidogyne javanica field. I'll talk about that whole testing process in a minute. But so bottom line for this trait and for the other ones that we looked at is we looked at a wide range of not only recent cultivars or carrots that are available to growers, but also heirloom cultivars, land races, which is carrots or any crop from distant places, more locally grown around the world, and that includes other types of local varieties. So when you're growing carrots of these diverse sources, like I said, most of the time you're going to see susceptible carrots like the one on the right. Yeah, in some cases, we were fortunate enough to find some carrots in the group that were resistant. And so this work was very much dependent on collaborations that we established with Phil Roberts at University of California, Riverside, and Bill Matthews working in his lab, where Phil and Bill have inoculated infested fields at three locations in California, Kearney, Tustin, and Coachella, with these nematodes that I listed in the last image. More recently, Bao Lam Hun has entered the picture as Phil has retired and Bao has come on the project, so we're continuing to work with Lam. So we did most of the warm climate nematode work in California at these three field sites. More recently, Phil has initiated a collaborative effort with Tim Waters at Washington State University, where there's some evaluation of carrots in a field infested with Meloidogyne hapla, and the cooler climate nematode. So that's one way to screen for nematode resistance, is to be fortunate enough to find a collaborator like Phil Roberts and Bao Lam that have infested fields to test for resistance. Beyond that, Phil and his group and now Lam are doing some greenhouse testing of soils infested with nematodes in the greenhouse at UC Riverside. And also we've started some work here, particularly with a grad student, Kevzer Rosell, looking at evaluating nematode resistance with soils infested in pots in the greenhouse with these nematodes. As I mentioned before, we looked at lots of different carrots. This is another picture of another carrot that is diverse in its nematode resistance. The process involves growing out these carrots, pulling the ones out that have the most resistance. Phil and Lam sent the roots back to me. We produce seed on them and go through the cycle, sending seed for the next year and continued through the years. So far, we found genetic sources of resistance, not only from Brazil, I mentioned Brasilia, but also Syria, several places in Europe, China, Australia, South Africa, and Tunisia. So there is nematode resistance in plenty of different cultivars or old land races, heirloom varieties, local varieties of carrots. What have we done with those? Well, as I mentioned before, Phil sends the carrot roots back to me. We produce seed on them and start breeding for this resistance, intercrossing the resistant carrots and developing resistant stocks. So here you see some nematode-resistant carrots that are being released. On the upper panel, upper left, all those with a prefix B are from Brasilia with different shapes and different genes for resistance. And also below that, some sources of resistance from Cape Market, South Africa, from Homs, Syria, from China, and from Europe. On the right, you see some carrots improved for fresh market carrot production with the long, thin carrots, 2306 in particular, being of some interest for baking baby carrots, which is primarily grown by the large-scale organic growers. But that 2306 carrot, which is very nematode resistant, when intercrossed with the two carrots below, which are also nematode resistant, gives some very promising cultivars for hybrids for fresh market carrot production. So these are some of the so-called roots of our labor, as it were, for carrot improvement for nematode resistance in the CIOA project. And then see, these are some, in the end, we're interested in moving these nematode resistance genes into more widely grown carrots. That's where those examples of the last slide come in. And these are some examples of some experimental hybrids from my program for fresh U.S. fresh market production, upper left, and for baby carrots on the right. Down below, just as a reminder, this is what the susceptible check looks like in our nematode screening fields. This is the Imperator 58, an old OP used in the U.S. that we grow roughly every 3 to 5 row plots in the field just to keep track of the intensity of nematode attack. So this is an example of carrot improvement for CIOA project for nematode resistance. Moving on to, and we've been quite successful in that process, and we've released quite a few carrots to the seed companies for improving carrots in their hands, because they're really the producers of the seed from the project more than we are. Moving on to the second disease of interest, alternaria leaf blight, we come to a foliar disease where this disease is typically scored on a 0 to 5 scale, where 0 would be disease-free. I've never seen a disease-free carrot plant under heavy alternaria infestation. Typically, the scores don't get much below 1.5 on this 5-point scale, and you can see what a score of 2.5 in the left and 5 on the right look like. So it's a very important disease and regarded both in the U.S. and globally as the most important foliar disease of carrots. And when the disease is in the area of the grower where carrots are grown, yield losses typically range from 28 to 75% due to reduced plant growth, so carrots that are undersized for sale, or weak tops, which translate to carrots that are lost when the mechanical harvesters that are typically used go through to pull the carrots out of the ground. And as I mentioned before, there's no complete resistance. I didn't mention it for the back on the nematode resistance, but we were pleasantly surprised to find some very strong nematode resistance in carrots. We've not been so fortunate to find strong alternaria resistance, but there are quite a few sources of partial resistance. So we have some carrots that we are releasing with the CIOA project with scores of 2.5 or so on that 5-point scale, which certainly I'd like it closer to 0, but that's a fairly good score in a field that's heavily has a heavy infestation of Alternaria dowsii. So the process for screening is similar to the nematode resistance. In this case, we're in fields where there's either a reliable natural occurrence of Alternaria dowsii, which is the case in central Wisconsin. But in some cases, we've actually inoculated with Alternaria dowsii as well to step up the intensity of the disease. Typically, you don't get a big step up, but it's been done. And we see resistant checks having scores of 2.5 to 3. Something like Bolero is a pretty good resistant check. But many in the field have scores of on the high end of 5, which means pretty much totally ruined tops of the carrots. Collaborators on this project include my colleague down the hall here in Wisconsin, Erwin Goldman, but also Mary Ruth McDonald at Guelph University and Jas Sidhu at University of California. Interestingly, we find the nematode ratings on all 3 locations to be relatively similar. And in the process of the CIOA project, we've got 6 entries out of the roughly 75 breeding lines in the CIOA project that have consistent ratings that are comparable to or more resistant than resistant checks. And these are not uninteresting entry numbers that we use in the project, but there's 6 very different carrots. The R ones are red, the others are orange from different parts of the world in terms of the origin of the Alternaria resistance. So we've made some good progress with Alternaria resistance, but again, we are not getting close to that complete resistance level, and I don't expect we will anytime soon. Moving on to the third disease that we've looked at, cavity spot is an important disease, and just to show you a couple of examples of cavity spot, it's usually not significant disease immediately after carrot harvest, but with any level of post-harvest storage, these spots on the surface of the carrot that don't look that could be peeled off, often on a freshly harvested carrot, only grow larger and really deplete the quality of the carrots with time. And so that post-harvest damage has made cavity spot be regarded as the most important disease in post-harvest cold storage, where that is done as often it's done for organically grown carrots that are local for local markets. In this case, again, we used infested fields that cooperators like Lindsay Dutoy, Mary Ruth McDonald, Jess Sidhu had either, in the case of Mary Ruth, naturally infested Pythium infested fields, whereas Lindsay and Jess had infested the fields with different species of Pythium. And we evaluated carrots in the CIOA project and other sources for cavity spot resistance. The photo on the right are maybe not too obvious, but on the upper part of the picture is our photos of the 7738 carrot, which there's some black spots that may not be too obvious. You are not cavity spots, but rather just specks of soil on the upper set of carrots, whereas in the lower carrots, which is a susceptible carrot, you'll see a lot of black spots, and in that case, that is the beginnings of that Pythium cavity spot. And and you see in the upper left some close-ups of the cavity spots showing up after some level of storage. And so that's what the selection process involves, is growing carrots in infested fields and then evaluating resistance in the plots from those fields. In the lower left are two carrots with cavity spot resistance, where 7738 has been a standout source of resistance. In that case, it's the carrot we stumbled into. We didn't really do any long-term selection for it. It just happens to have quite good cavity spot resistance. Also in the lower left is actually a nematode-resistant carrot I mentioned earlier that also has some cavity spot resistance. So looking at our progress in cavity spot resistance, 7738 is a line that has been released to the seed companies and growers for their use in breeding. And we have several other carrots in the CIOA project that have strong resistant ratings. And these are being advanced in the project, and some of those will also be made available. Interestingly, the scores we get in Washington, California, Canada, with the cavity spot trialing by my collaborators I mentioned earlier, give you very similar scores. So that's where we are with the three diseases that I mentioned. Moving on to another very important trait, weed competition is certainly important for both organic and conventional growers, because carrots are it's all from seedling, and it's all very slow compared to weeds. And that's true for both conventional and organic, but for organic, it's dollar-wise can be much more important because of the inability and non-application of herbicides. Hand weeding can cost up to $10,000 a hectare for carrots, so it's not cheap to control weeds if you haven't otherwise managed them before the weeding crew has to come through. So we've been looking for genetic variation for several traits we thought would contribute to better competition with weeds. So things like carrot seed germination, early growth, early season vigor, plant height throughout growth, leaf area, possibly allelopathy, canopy architecture. These are things that have been looked at in other crops and we've looked at in carrots. And several of these traits have been applied for as better ways to have crops compete with weeds. In the case of sweet corn, wheat, barley, and now also in carrots. For most crops, there's not a lot published on weed competition as there is for disease and pests, but there is some on carrots, and we've added more to that literature base. So again, we were doing our work with the CIOA project collaboratively with Jed Cahoon, University of Wisconsin here in central Wisconsin, and also Sarah Turner was a grad student doing some work here in Wisconsin. And in the case of Jed's work, the fields were infested with weeds to look at the competitiveness of carrots to the weeds. But since carrots grow closely to each other, carrots growing to each other also provide some useful information for weed competitiveness, what Sarah looked at. So here's a view of our field in Southern California and our winter root nursery, and you see some very stubby tops on some of these carrots. They have plenty of root size, but not much carrot, not much, lots of roots, but not much tops to see. And so very poor weed competition for some of these. These all, of course, were controlled for weeds. We didn't let the weeds grow. You see, in addition to the almost no tops on some of those carrots, quite a bit of variation for top size. And so we tapped into this variation for the project as well. Carrots are grown at high density relative to many tops, and so tall tops really are required to replace hand weeding when there's a lot of weed competition. And Jed Cahoon demonstrated this in his project. In Sarah's project, she was looking at the range of top height, and there's at least a twofold range for top height. Here you see a couple of examples between two breeding lines, and that's another angle on looking at improving carrots for weed competition that we're looking at in the CIOA project. So where have we come in the project thus far? We've looked at amongst our roughly 75 carrots in the carrot CIOA project. There are 9 entries with relatively tall tops. And as Jed Cahoon had demonstrated, that variation amongst those tall tops versus short tops carrots weed loss of productivity due to weeds ranged from 38% on the tall tops carrots to 87% yield loss on the short tops carrots. So top size is truly important, and there's variation. We're tapping into it and releasing carrots with tall tops as part of the CIOA project. So now we've moved through the traits that we've addressed with more typical breeding approaches for pests, disease, and weed problems that organic growers face. But looking at soil nutrients is something that others in the project have looked at because there is a significant difference between organic and conventional production systems in terms of the soil type and nutrients in the soil. A lot of this work on other crops is focused on nitrogen availability, but also there's been a fair bit of work on mycorrhizal colonization looked at in a few other crops. And for CIOA, the team that looked at the organic versus conventional comparison for soil nutrients looked primarily at mycorrhizal and endophyte research in the CIOA project. Now I'll discuss that in the next couple of slides. So in this case, carrots were grown in organic and conventional fields, or in some cases in greenhouses with soils from those sources. And the University of Wisconsin team, including Aaron Silva, Zach Friedman, Julie Dawson, have looked primarily at mycorrhizal fungi and their relationship between carrot growth in a couple of different cultivars grown in organic conventional conditions, both the effect of the fungi on carrot growth and also looking at some of the root exudates that come along when carrots are grown and see how those relate to carrot growth. The other project looking at soil nutrients was Lori Hoagland's, looking primarily at taproot endophyte composition as it varies, again, in organic and conventional soils with several different cultivars grown in the greenhouse primarily. So this slide summarizes their work, and to be very honest, to fully cover this would really take a whole nother separate webinar, so I'll be brief and cover it specifically for how it relates to the breeding part of the CIOA project. But with the Wisconsin group looking primarily at the mycorrhizal fungi, there was a comparison between heirloom open pollinated carrots and modern hybrids to look at the effect on carrot growth, and there were significant differences when grown under the same mycorrhizal conditions. And more recently, some work following up on that to look at some of the root exudates that may mediate those carrot microbe interactions. And that work is still underway, but there's some indications that this could be a precursor to predicting some carrot growth inorganic soils versus different organic soils versus conventional, based on that as something to evaluate in a carrot evaluation and breeding program, or at least a trialing program. In Lori Hoagland's project, soils were looked at with different organic matter abundance and microbial biomass and root endophytes. In other words, those microbes in the carrot root were evaluated for their diversity. And the grad student working on that project observed significant differences in those that endophyte composition that may be a harbinger of what carrot productivity is ongoing. There's ongoing work then to evaluate what that endophyte variation in composition, how it relates to what growth, similarly with the mycorrhizal fungal work. So at this point, we don't have a specific breeding plan for using this information, but as this type of research moves forward, I predict that these microbe interactions can become a trait that will be evaluated by carrot breeding programs. So not as much a report for this particular trait, but just to say that there's some important work that's come out of the CIOA project and more is underway. Finally, moving back to some specific traits where there's more conventional breeding underway, is looking at consumer traits, again, because of the fact that whereas obviously consumer interest and preference is important for both conventional and organic growers, but for organic growers, there's much more interaction with the consumer. And there is a lot of variation for consumer quality in the form of flavor, texture, nutritional value, and novelty. And this can have a very significant impact on what consumers are going to buy. So the CIOA project has looked at breeding carrots with variable color, flavor, and texture to better address consumer interests and needs. So if we look at just carrot color, we see this huge variation amongst different carrots for color, and it's a widely variable crop. So we've tapped into that variation to develop some new breeding lines with a wide range of colors based on these carrots. The one in the lower left on this picture, a circle of yellow, orange, and white carrots, is landraces from around the world. There's one purple in the group. But there's a number of different colors. The primary ones are yellow, red, purple, and orange, as well as white with no pigments. And so a lot of variation for carrots in color and also in shape. That's important. So looking at some of these variables, that same variation you see for color is also borne out when we look at things like texture and flavor. And the project has gone beyond color to look certainly at those variables, because in the end, consumers really pay attention to flavor and texture in making their preferences and coming back for second purchases of a given carrot. And the primary collaborators here have been Michaela Colley, Organic Seed Alliance, and Julie Dawson, University of Wisconsin. So we've released several carrot lines. The upper left are a couple of red carrots, 6220 and carnelian, a yellow and a purple to the right, a couple of others on the bottom, and some of the variation for color down below in breeding lines that some of those will be released that vary for color. As I said before, flavor is critically important. There's a lot of evaluation of flavor that we've had underway. There's also a growing body of data for the genetics of flavor and some of the genetic markers we can use to improve the crop without actually maybe tasting them all. But at any rate, we do continue to taste them in panels like the one you see in the lower left here. This evaluation of genetic variation of flavor and texture has been a significant component of the CIOA project. In the 75 or so carrot breeding lines in the CIOA project, all have been improved for flavor, some further than others. And with this work, we've generated a large body of published research on the carrot carotenoid, or orange, yellow, red pigments, and purple pigments, anthocyanins, genetics with the project underway. We've got some significant work underway for variation in carrot texture, which also varies significantly, and looking at post-harvest storage of the crop. So I've gone through in pretty quickly, but just to cover the range of things in the CIOA project, I've gone through the traits that we've looked at in the CIOA project. And just to summarize and recap, there is a broad range of genetic diversity in carrots that provides growers with disease and pest resistance and weed competitiveness that can be very valuable to them when they are able to tap into this genetic variation. In addition to that variation for field traits, another field trait is the interaction with the soils of organic production. And that's where the root microbiome teams at University of Wisconsin and Purdue have been very productive in generating some new information that demonstrates some of the differences between conventional and organic production, making this a potential trait of interest and certainly an area of continued research for organic breeding and improvement of carrots. Along with these traits and this variation for growers, variation for consumers, both that visual variation for color and shape, as well as the flavor, organoleptic and texture variation, and nutritional quality, which goes along with color in carrots. There's many opportunities to go beyond what we've tapped into, but we're releasing a fair bit of variation with CIOA releases for the project as well. And again, developing genomic tools to help those breeders that have access to these tools to speed up the breeding process. One more topic I'll cover briefly before I wrap things up here is along the way, we evaluated 36 breeding lines for 4 years in 2 locations under both comparing productivity traits under both organic and conventional management. And the results of that work published by Michaela Colley as the lead author demonstrate that management system did have some influence on carrot performance, but no clear trends that applied across genotypes or conventional versus organic environments or growing locations. The results indicated that breeding in either conventional or organic has potential to address organic traits, but the evaluation for weed competitiveness in particular is much better to evaluate in the organic production system. And the final conclusion from that work is that the genetic improvements in conventional systems have been delivered and will continue to be delivered to organic growers, but selection in organic systems may speed up the process of evaluating the best growers under these disease, pests, and weed competitive situations. At least testing cultivars under organic conditions is recommended to ensure that the cultivar being tested is optimal for organic systems. So again, I'm not going to recap this too much about up for time here. We've identified new gene sources. We're releasing carrot variation in our germplasm that's available to seed companies, growers, and of carrots and researchers. And we've made significant projects in understanding the genetics of some of these traits, but also realizing that there's a lot more that can be done to improve any of these traits that I've talked about today. We really just tapped into that genetic variation for carrots in all of these areas. But with this work, we anticipate that future advances can be anticipated from our work. So with this, I'll say thank you and again, remind you that it's been important to have this funding input, but also to have your interest in what we're doing to help us get things out to you as the growers. Thank you.
Thank you very much, Phil. That was a great presentation. And I just wanted to mention before we start the question and answer that we have a website for the project at eorganic.info carrot improvement. And there you can find the publications from that project, as well as any kind of news and upcoming events and that type of thing, as well as some carrot photographs. So feel free to check out the website, and I'll type that into the chat in a moment when Phil is answering some questions. So with that, we have some interesting questions coming in, so I'm just going to get started here. Does your breeding program prioritize mechanical harvest? You hadn't mentioned that at all.
To some extent, we do. Mechanical harvest translates to carrots that, by definition, at least with most of the mechanical harvest for fresh market carrots, is pulling the carrots out by their tops. And so that translates to strong tops in addition to tall enough tops, but the strength of carrots to the tops of the roots varies. I can't say we prioritize it so much as we pay attention to it because it's significantly variable under Alternaria attack. And even if you're growing carrots and loosening them up with a fork before you lift them by hand, you're usually pulling them up by the top. So top strength is important. That's not a very clear answer, but we do pay attention to top strength attachment.
Okay. Let's see. Here's another question. Bejo had a non-hybrid Nelson that is unsurpassed in quality. They have dropped it. Any chance of reviving Nelson or coming up with something that's equivalent in quality?
You'll have to talk to Bejo about reviving Nelson. I'll transmit your interest, but you'll probably get more effective response to do it directly. But we certainly are paying close attention to flavor. So I guess on the question of flavor, I guess when I started breeding for improved carrot flavor, I thought I could come up with a definition of the best flavor. At this point, having tasted an awful lot of carrots, I find out there's some parts of carrot flavor that most people agree on, but some that people don't. So the things we look at are sweetness, which pretty much everybody agrees carrots better, sweeter carrots would be better. The other variable is thing we call harshness, which is a turpentine type of flavor. It's caused by terpenoids in carrot roots, same compounds in pine needles. The more you have, the more carrot flavor you have. Some people like a stronger carrot flavor. That means more turpentine carrots. Some people prefer less. There's a wide variation there. Offhand, I don't recall we've looked specifically at Nelson, but I'm familiar with it. It is a good tasting carrot. Whether everybody on this call agrees it's the best one, I'm not sure. I'm sure many will. But yes, we are looking at particularly good flavor and now more recently good texture.
Okay, let's see. Oh, this sounds like a dangerous question. Is it true that the Dutch bred carrots to be orange?
That is a dangerous question. My take on that, based on the historical information, which is primarily based on looking at works of art in Europe, also a little bit based on written information, but not so much on the orange question, is that orange carrots came into and were grown in southern Italy and southern France and widely in Spain before they got to the Netherlands. So I would say quite certainly the Dutch marketers of vegetables were the ones that got orange carrots well marketed, like they've done with many other crops. Whether they were invented by somebody in Holland, I'm not sure. But these things develop slowly. The records are poor. New work of art that has carrots in it might give us more information, but that's my take on it as of today.
Okay, that was a very diplomatic answer. So we don't want
I've
to
learned I can
risk
get in
our
big arguments on that question.
right,
So
I'm sure.
I
Yeah,
don't.
we don't want to bust any myths there. Okay, so can less nematode-resistant cultivars be grown successfully in biologically well-managed soils? Must the whole resistance burden be borne by genetic
Uh,
resistance versus management?
uh, so um, I'm not sure. So One thing I'll note is that it has been observed that organic soils tend to have less nematode attacks on them if you're growing carrots on otherwise organically grown soils, not just for nematicides, that there tends to be less nematode attack, but they do attack certainly very much under organic inorganic soils as well. Um, I guess I'm not positive about what the question is. There's no downside for carrot productivity that comes along with nematode resistance. Um, so, I guess I'm not sure. Alice, do you have any insights as to whether I'm answering the question
Yeah,
right?
well, it sounds like it. Yeah, it sounds like it. If not, feel free to type in more detail
Yeah,
there.
and
So
I
if you didn't address exactly what you meant. So
and yeah, and
yeah.
I'll mention as an aside that interestingly, we don't see any significant reduction in productivity as a function of including disease or pest resistance, flavor, things like that. There may be a little bit of down yield production with orange versus white carrots, but that's minimal.
Okay. Yeah, feel free to type in more questions if you have more questions about that issue. Next question is, how does the tall top selection impact carrot flavor and nutritional value? Does the energy sent to the carrot green impact the nutrient density on the root?
Uh, to what we've seen, there's no effect. Probably the big effect is some farmers, including organic growers, have too much nitrogen in their soils, or too much from the standpoint of what's necessary for carrot production. I think that growers of crops like carrots, root crops, like to see big tops on their plants with the guess that the roots and bulbs and tubers underground must be growing very well as well. Um, that is not necessarily true. You can have too much nitrates available that give you big tops, and with not so much better compete with weeds because when there's all that extra nitrogen, the weeds do better as well. But anyway, just to point out that from a crop management standpoint, you can boost top size with different forms and amounts of nitrogen fertilizers and other fertilizers. But there's not any significant change in flavor or color or things like that, other than the fact that you got the plant growing a lot of the tops and putting more resources there. So in some cases, the roots tend to be smaller with too much nitrate addition. But if you're managing things well, and I'm assuming all our listeners here are, you will not find any difference in the taller tops versus the shorter tops from anything we've looked at.
Okay, great. Let's see. Could you possibly expand a bit on the interaction between microbiology and organic heritage varieties versus modern carrot varieties? Did I understand it correctly that the modern varieties have less of a network?
Right, that's been the interesting discovery. So this is with, so just to back up a little bit, when I talked about nematode and alternaria and cavity spot resistance, we are looking at literally hundreds, maybe 700 to 800 different diverse carrots to come to conclusions. These these microbial interaction experiments are much more limiting in what can be done because it takes a lot more research effort to get that data. And so the studies have been relatively small. We started, my collaborators started with small numbers of carrots that included some more heirloom types or included some modern cultivars. Some include also, I didn't mention it much, but included some disease resistant versus susceptible carrots. So maybe 6 to 10 different carrots in the study. And so based on those relatively scant data, that's correct that the heirloom varieties tended to respond better to those microbial situations in organic soils than do the more modern varieties. So some of that landrace and heirloom heritage that those cultivars retain shows up when grown under organic production in these not too large experiments. And please don't get me wrong, the collaborators worked very hard to get this data. They put a lot of work into this. It's just something that at this point hasn't got to the point of large scale screening. But it's been some very interesting conclusions based on those small numbers that it does look like what went on before the conventional management systems were in place in those heirloom varieties, provides a better genetic profile for carrots grown in organic production in the terms of the microbial interactions.
Yeah, so I guess the follow-up to that question was how do organic varieties compare? And I'm not sure if he meant organic, like carrots that were actually the seed was bred
Yeah,
in organic production.
yeah, so we did not compare seed grown under organic, seed produced under organic or conventional conditions. We only, I think I'd have to double check, but I think in all case, we maybe did have some comparison of something like, we may have had Bolero both from organic seed production and conventional. But anyway, we didn't compare that very extensively. Not enough to give any results.
Okay, we have the breeder on who's been interplanting carrots with potato and tomato breeding and wants to share his insights. And then I guess, well, I have another follow-up question after that, but can people contact you to share their insights and how could they do that?
Yes,
Mhm.
I'm sorry I didn't include um, an email address, but yes, I would welcome any interaction, questions, follow-up things that make, suggestions. Yes, you can find my address, I think, pretty easily. I have both the USDA email and a University of Wisconsin email address.
Yeah,
Either
and I
one
can
works.
attest he answers both of them. So
Yes, I do answer both of them. Yes.
yeah. Okay, and then also, I guess, kind of a follow-up for that. I had a question about where people could people also contact you if they're interested in some of your breeding material.
Yes, for the release lines, as a USDA project, we give away seed samples, although sending seed internationally today is challenging given current situations. But we do have seed available. Don't expect a lot of seed. You may only get a couple hundred seeds. But if at my website we have a list of all the released carrots, both from this project and other projects, and if they're listed there, and you have a U.S. address, I can send you seed samples of those. If you're international down the road, I hope to be able to, but not as of today.
Okay, let's see. Um, oh yeah, have you published anything on the microbial associations of heirloom versus modern cultivars?
Yes, I only included the names of the senior author with an et al. On that one slide I had talking about the root microbiome interactions. And there are at least three, well, I don't know, at least five publications on that topic and more in the works. So again, if somebody contacts me, I will definitely provide you with links to those references and also those researchers that did the lead work on the actual root microbiome evaluations are members of the project, primarily Lori Hoagland, Julie Dawson, Aaron Silva, and Zach Friedman as the lead PIs on the project. I'm sure they'll be able to respond to questions.
Yeah, and that article ought to be on the website now. And if it isn't, we can check afterwards and make
Right.
sure that it
Yes.
is, but
Yes.
it should be
Yes.
there. So
Yeah.
I put the website link into
Ah.
the chat so
Yeah. Yeah. Thank
people
you.
can go there and check it out afterwards. Let's see. Here's an interesting question. You haven't really addressed this so much, like the characteristics of the different colors of carrots, but we had a question about whether there are any benefits to the color orange. Again, a question from the
Hmm.
Netherlands. So
They're much preferred by Dutch consumers, but no. People, consumers today expect orange. And so that's looking at all consumers. Now, I'm sure many consumers on this presentation have more refined and diverse tastes that that may bring you to be more curious about the novel colors. A colleague, now retired extension specialist in Washington State, had some purple, red, white, and orange carrots that he provided at the Pike Place Market on Saturdays in Seattle. And he found that when he had slices or sticks of these different colored carrots, that kids would try all the different colors and give them their responses. But he said some adults would get that purple carrot by their mouth, and he said, put it down and say, I can't eat that. That's not the right color. So depends on your curiosity and willingness to try something different. It can be an attraction, could be a distraction, but we're viewing it as an attraction and continuing to breed the novel colors and shapes.
Do you have a favorite particular color carrot or particular favorite variety? Mm.
It's like asking which of my kids is my favorite.
That's
No,
true.
I don't.
No,
So,
okay. Okay, let's see if we have any more questions here. We have some more information from the plant breeder who's on saying that he's evaluating carrots with characteristics like weed competition and has photos and
great.
yeah, so that would be great if you would like to contact Phil and share that with him. I'm sure he'd be interested. So
Yes,
um, yeah, let's see. Oh yeah, okay. Um, let's see. Do the different colors have different nutrients?
very much. Yes. And what you see is what you get. And you look at this circle of carrots in the middle of this picture, upper top part of the picture. Darker orange translates to more nutritious. Yellow translates to lutein, which is good for eye health, but it's not a vitamin A precursor. We don't have much vitamin A deficiency in the more developed part of the world, but for what it's worth, that yellow pigment is important for people that might be subject to macular degeneration. Color is a good antioxidant, and those purple orange carrots in the bottom, even though you can't see the orange in the outer part of the root, that in the cross sections, they also have carotenes in that part of the roots. It's not only purple. So yes, so the different colors very much demark different human nutritional needs, which is very interesting. But all the colors are more nutritious, and for that matter, even white carrots are good for you because the fibers are very good for all carrots.
Yeah, so it sounds like it'd be great to grow a diversity of carrots and get all the
It,
different nutrients.
yes, that's always my suggestion.
Okay, I think we've come to the end here. Oh, we have somebody who wants to know. Oh, okay, just a couple more. We have a little more time, so I'm just going to finish up the questions here.
Sure.
Okay, let's see. Oh, yeah, somebody wanted to know if you're ex accepting graduate students.
At this point, not, but some of my collaborators may well be. But so I can, if refer to this presentation, and I'll work with you to see if I can find one of my collaborators that might be
Okay,
looking for new grad students.
yeah, and then we have a question about whether there's a website that where you can see the different nutrients by color. I know that on our
Hmm.
website we haven't updated the carrot
Mm.
catalog for a while, so I think
Yeah,
we don't
I,
have enough information about that just yet. But
yeah, but
is there
I've
somewhere
got
where you can find that? Mm-hmm.
I've got a handout page I'll send to you, Alice, that
Mm-hmm.
it's called Pigment Power.
Okay, yeah,
And
it sounds familiar. I'm gonna have to
yeah,
check and see if
I'll
that's already on there. It might be, but
it might be.
yeah.
I've had it out there a while, but it talks about what the different colors refer to nutrient wise. So again, I talked about yellow versus for macular degeneration, red with some of the antioxidants for cancer prevention,
Yeah, so check
Yeah,
the website there. It's eorganic.info/carrot improvement. And then Paul very helpfully sent a link to that on the
okay.
ARS website. So thank you
Yeah,
very much, Paul. If you look in the
yeah,
chat, you can see that Paul very kindly sent that to everyone. So
yeah,
yeah, whether or not it's on our website yet, which I think it may be, there's a link to it right there that you can click on. So
yeah.
thank you very much. And yeah, I think we've come to the end here. So I'd like to thank you so much, Phil, for this really
Well,
interesting and informative presentation. And we welcome everyone who's on right now to come to Phil's next webinar next month, which is all about carrot history and domestication and diversity. So that one is sure to be an interesting one. It's also going to be, I think, a little bit about Phil's travels around the world looking for carrots. So yeah. Any concluding words, Phil?
my concluding words are thank you so much for the opportunity to speak to you all today. I appreciate your interest and look forward to some continuing conversations with some of you. Thank
Okay,
you
thank
very
you.
much, Alice, for organizing this.
Oh, you're very welcome. Thank you.